So we're very pleased that the European Hematology Association picked our abstract as one of the 6 late-breaker abstracts tomorrow morning. So what we will present is the first-time clinical data disclosure on a phase 1 trial that tests a in vivo CAR-T construct targeting CD19 and CD20 in relapsed refractory non-Hodgkin lymphoma patients. This construct is codenamed LB2501. It's an in vivo CAR-T, which means we're actually injecting the lentiviral vector directly into the vein of the patients. And then from there, the patient's T cells will be transduced, and then the CAR-T cells will expand in the body. So essentially, we're moving the factory to the patient's own body to generate a CAR-T cell that serves as a therapeutic for cancer. And this will eliminate the need for waiting time, the need for apheresis, in which we collect the immune cells from the blood draw. Also eliminates the need for a 3-day chemotherapy called preconditioning regimen, that's lymphodepletion. And also what's important is that in this type of trial, the patient do not need to wait. They can get the therapy pretty much immediately after diagnosis. We're choosing 2 antigens, CD19 and CD20. The reason is there are 3 commercially available CD19 CAR-T therapies in the market, namely Kymriah, Yescarta, and Breyanzi. However, some patients relapse from those therapies because in certain patients, the CD19 antigen will be downregulated, or even there's escape or loss of expression of CD19. So if you target both CD19 and CD20, which are expressed on the cell surface of the cancer cells, then it's quite unlikely, or it's less likely at least, for the cancer cells to mutate to actually overcome both. That is why we picked both. Tomorrow we'll present is 12 patients in the ongoing phase 1. 6 patients received the first dose level 1, where we see minimal expansion and no response. However, in the 6 patients who were dosed in dose level 2, all 6 responded. So we have a 100% response rate. What's more remarkable is that out of the 6 patients, 5 achieved complete response, which means by PET-CT scan, there's no sign of the cancer activity anymore. This is very encouraging data, although early with 2.2 months of median follow-up. But this is the first report of clinical data for in vivo CAR-T in the field of lymphoma, and we're very excited about that. So Legend started 12 years ago, and we do have a commercial product named CARVYKTI, which is FDA indicated as a second-line therapy for multiple myeloma. In 2025, CARVYKTI was the #1 selling CAR-T therapy in the world, with nearly $1.9 billion in global sales. And it is the biggest CAR-T therapy in treatment of multiple myeloma. In terms of research and development, we have done research in broad categories, including autologous CAR-T, allogeneic CAR-T, and now most recently in vivo CAR-T. We want to bring CAR-T cell therapy into patients with solid tumor, blood cancers, and eventually maybe also autoimmune diseases. That's what we're doing. We're trying to leverage the power of the immune system and T cells to bring therapies to patients with unmet medical need. CARVYKTI was the first CAR-T therapy that shows long-term durability and also curative potential in late-line patients. In second-line patients, we're seeing superior PFS, overall survival, and also MRD negativity compared to standard of care. So we're making a lot of inroads as a new therapy. What also is exciting that's happening in the field of multiple myeloma is that besides CAR-T, there's also a category of T-cell engagers. There are multiple bispecific T-cell engagers that are approved by the FDA now. In fact, at this conference, we just saw this morning a presentation on TECDARA, which is TECVAYLI, that is a BCMA targeting T-cell engager, in combination with DARA or DARZALEX, which is a CD38 antibody. Again, you're seeing very good results compared to old standard of care. So you're seeing a lot of new development and also newer regimens with better durability and the better overall response. And we're very excited about that. We're just very excited in general about all the scientific breakthroughs and also advances in the treatment of blood cancers. Particularly, we're very interested and excited to see the early results of in vivo CAR-T in the field of blood cancers.